Hello and welcome to today's webinar. On behalf of Drug Target Review and Eurofins Discover X, I'd like to thank you all for attending today. I'm your moderator, Victoria Rees, Deputy Editor of Drug Target Review. Today's speaker will be Verena Albert, PhD, Lead Scientist for Cardiac Safety Sciences at Eurofins Discovery. Following her presentation, we'll be moving on to a live question and answer session where you can pose any questions that you might have to today's speaker. Please do remember you can submit your questions at any point during the webinar using the questions panel situated in the menu on the right. But without any further hesitation, may I pass over to today's keynote speaker, Verena Albert.
Thank you for this kind introduction and welcome to the webinar also from my side. Today, I would like to talk about cardiac proarrhythmic risk assessment and present to you the assays that we are offering here at Eurofins to investigate cardiac risks of new compounds using human-induced pluripotent stem cell-derived cardiomyocytes. First, I want to give you an overview of cardiac liability testing for drug development and explain to you how this area has evolved over time and give you an overview of the current regulations. I then want to explain the Cipa Initiative, which is a new and improved approach for cardiac liability assessment. And the main part of today's talk will be focused on in vitro phenotypic assays using human-induced pluripotent stem cell-derived cardiomyocytes to assess cardiac liabilities during drug development. And I will be focusing on two different assays, the multi-electrode array assay and the Flipper calcium transient assay, which are two assays that we are offering here at Eurofins to assess cardiac liabilities. So adverse drug reactions are one of the main reasons why new drugs fail in clinical trials. As you can see here from this pie chart on the left, almost a third of all drugs that fail during phase 2 and phase 3 clinical trials fail because of safety concerns. And this means these drugs, besides targeting their intended main target, they also target additional off-targets. And as a result, patients develop adverse reactions. And so this is why they cannot be developed further. The reason why this percentage is so high in such a late stage of drug development is because safety assessment is reserved for the clinical phase of drug development. And so you can imagine that this causes a big loss of both time and money for pharmaceutical companies. As a consequence, in recent years, there has been a strong increase of in vitro safety pharmacological profiling and in vitro phenotypic assays earlier in the drug discovery process in order to identify those drugs that have adverse drug reactions much earlier in the drug discovery process. If we look at what kind of safety concerns these drugs have that have to be withdrawn, you can see that cardiovascular toxicity is one of the major causes for failure of drugs during drug development. In addition, cardiovascular toxicity is also one of the major reasons for post-approval withdrawal of already released medicines. The reason why the cardiovascular system is one of the main systems affected by adverse drug reactions is because the heart is essential to keep the body supplied with oxygen and nutrients and to clear away harmful waste. On a molecular level, there are electrical signals that cause the heart to contract. These electrical signals, they arise from a group of specialized cells from the sinoatrial node, and the signal travels from the top of the heart, so from the atria, to the bottom of the heart, to the ventricles. And a brief change in the membrane potential across the cell membrane of the cardiomyocytes causes the contraction. And the change in membrane potential is caused by movement of positively charged ions such as sodium, potassium, and calcium through cardiac ion channels. And because the heart is so important for the body to stay alive, Only a very brief interruption of the rhythm or the function of the heart results in irreversible multi-organ damage and even death. And so for newly developed drugs, it is really important to avoid that they by accident also target cardiac function. The electrocardiogram, or short ECG, is a non-invasive and very efficient method to monitor the electrical activity of the heart. It has a very characteristic waveform which corresponds to the different parts of the contraction. It starts with the P wave, which represents the depolarization of the atria. It is then followed by the QRS complex, which represents the depolarization of the ventricles. And then in the end, it ends with the T wave, which is the repolarization of the ventricles. And a change in the shape of the waveform can indicate that the patient has an arrhythmia or suffers from another heart condition. One important measurement is the QT interval, which is defined as from the start of the QRS complex to the end of the T wave. And this is basically the time that it takes for the ventricles to depolarize and repolarize again. And a prolonged QT interval can lead to potentially fatal arrhythmias, such as torsade de pointe or short TDP. And the hERG ion channel is very important for cardiac contraction. hERG stands for human ether A-GO-GO related gene, and it is a cardiac potassium channel. It is very important for the repolarization of the cardiac membrane by conducting potassium ions out of the cardiomyocytes. And why it has been important for cardiac safety evaluation is because it is known that the hERG channel promiscuously binds a wide variety of structurally different compounds, and its inhibition causes the delay of ventricular repolarization, and this results in QT interval prolongation. And what is important to remember is that QT interval prolongation can, under certain circumstances, lead to fatal arrhythmias, such as TDP. The importance of the hERG channel for cardiac function has been noticed in the late 90s and early 2000s because patients who were taking these drugs suffered from cardiac arrhythmias and other cardiac problems. And what has been realized later on was that all of these drugs are potent hERG blockers. And the reason why these drugs were released to the market was because arrhythmia and TDP frequencies are too low to be detected in clinical trials. And because all of these drugs were blocking the hERG channel, the FDA introduced some guidelines in the year 2000 that required that in the future, all new drugs need to be tested whether they block the hERG channel and prolong the QT interval before they can be released to the market. And so in that case, hERG inhibition and QT prolongation was actually used as a surrogate marker to test whether a drug has a risk for cardiac arrhythmias. These guidelines were very efficient in preventing new drugs being released to the market that had cardiac liabilities. As you can see in this table here, after the year 2000, there was only a very small number of drugs that had to be withdrawn from the market because they had cardiac safety issues. But even though these new regulations were very efficient, you have to remember that hERG inhibition and QT prolongation does not necessarily mean that the drug will be a cardiac safety problem. And so consequently, what you can say is that an hERG and QT only focus can actually have a negative impact on drug development. What the FDA guidelines assumed was that every drug that blocks the hERG channel would behave like is shown here on the left side. So you have an hERG channel block, which results in QT prolongation and as a consequence, arrhythmia such as TDP. But by having this view, cardiac safe drugs might actually falsely be identified as cardiac unsafe. And the reason for this is because a single target assay, such as the hERG channel, cannot predict how the drug interacts with several different targets. And what has been observed is that when a drug, in addition to blocking the hERG channel, also blocks other ion channels such as sodium or calcium channels, you have a balanced ion channel block, which still causes QT prolongation, but it does not lead to arrhythmia. And here you can see a list of 3 drugs which behave exactly in this way. We have Prozac and verapamil, which both block the hERG channel, but in addition, they also block the calcium channel Cav 1.2. And then on the other hand, we have ranolazine, which also blocks the hERG channel, and in addition, the sodium channel Nav 1.4. But all of these drugs are cardiac safe. So for this reason, there is a need for an improved cardiac safety evaluation. And this is where the Cipa Initiative comes into play. And Cipa stands for Comprehensive In Vitro Proarrhythmia Assay. The Cipa Initiative was initiated in 2013 by several different agencies and organizations. And Eurofins has been heavily involved in establishing the guidelines of this initiative. The Cipa Initiative is an effort to improve the proarrhythmic risk assessment by focusing on 4 main points. So first off, you should test in vitro the effect of new drugs on multiple ion channel currents. And so this means besides assessing the effect on the hERG channel, you should also test how the drug affects calcium or sodium channels. After that, you should use this information and perform in silico computer modeling in order to predict the clinical risk for arrhythmias to occur depending on the results that were obtained in the first point. Then, as a third step, you should check for unanticipated effects in human stem cell-derived ventricular cardiomyocytes. And you should perform phenotypic assays for that to see how the drugs affect the cardiomyocytes. As a last step, you should perform phase 1 clinical trials and focus on in vivo ECG biomarkers. And this allows a more integrated approach to assess for proarrhythmic risk. Here at Eurofins, we follow the Cipa initiative and we offer assays that support several points of these 4 points. First of all, we have our cardiac ion channel services, where we can measure the effect of drugs on all the 7 main cardiac ion channel currents. In addition, we also have the cardiomyocytes assays, where we perform phenotypic assays such as the MEA assay and the Flipper calcium assay to assess the effect of the drugs on the cardiomyocytes. For the remainder of this presentation, I will now focus on these in vitro phenotypic assays using the cardiomyocytes. I will explain the background of the assays and show some example results. Human-induced pluripotent stem cell-derived cardiomyocytes are a very suitable tool to assess cardiac liabilities in vitro. The reason for this is because these cells are derived from skin or blood cells that have been reprogrammed back into an embryonic-like pluripotent state, and then they have been differentiated into cardiomyocytes. And so for this reason, they can be produced in large quantities, and that makes them suitable for in vitro testing. These cells also express all cardiac ion channels that you can find in cardiomyocytes in the heart. They generate action potentials and calcium currents, and they display spontaneous contractile activity, as you can see in the video here. And with these cardiomyocytes, we are performing in vitro phenotypic assays. Unlike in vitro pharmacological profiling, which is target-centric, meaning we overexpress one single target of interest and see how the drug affects this target, in an in vitro phenotypic assay, we are not limited to known adverse drug reaction causing targets because the readout is the phenotype, so the behavior of the cells. And this complements the in vitro pharmacological profiling very nicely because the phenotypic assays provide a more physiologically relevant readout, you can capture the interaction of the drug with several different targets, but it is still in vitro and there is a high throughput format possible, but it is much closer to in vivo models. So which are the phenotypes that we are assessing in the cardiomyocytes? We focus on two different phenotypes. In the MEA assay, we are measuring the action potential, and in the Flipper calcium flux assay, we measure the calcium oscillation. And importantly, drug-induced changes in these phenotypes indicate that the tested drug is likely to negatively affect the heart function. The multi-electrode array, or short MEA assay, is an electrophysiology-based technique where microelectrodes are embedded in the surface of the well where we are culturing the cells in order to measure fluctuations in the extracellular field potential, which is generated from the spontaneously beating cardiomyocytes. And importantly, the recorded field potential signal is analogous to the recorded electrocardiogram from patients. And changes in the field potential waveform can actually be used to predict the identity of the ion channels that are impacted upon drug exposure. Here you can see an overview of the MEA assay protocol that we use. We obtain the cardiomyocytes that are already differentiated from Fujifilm cellular dynamics, and we plate the cells directly into the plate that contains the microelectrodes. After culturing the cells for 4 days, we replace the maintenance media, and then we incubate the cells for another 1 to 4 hours at 37 degrees Celsius. After that, we record the baseline trace and we add the compounds. We incubate for another 30 minutes at 37 degrees Celsius, and then we record the post-treatment trace. We are evaluating 3 different parameters in the MEA assay. First of all, we are measuring the spike amplitude. This is a sharp transient spike that is associated with sodium ion influx and membrane depolarization. Second, we determine the beat period, which is the time between 2 spikes. And third, we also measure the field potential duration, which is the time between the initial sodium spike and the maximum of the potassium repolarization. And this measurement is analogous to the QT interval in the ECG. So here you can see some example traces where we treated the cardiomyocytes with different ion channel blockers. In black, you can see the control traces, so the traces where the cells have been treated with vehicle. And in red, you can see the traces of the cells that have been treated with the compounds. When we treat with a sodium channel blocker, and in this case we used mexiletine, you can see that the spike amplitude is strongly decreased. When we treat with a calcium channel blocker, in this case we use diltiazem, you can see that the field potential duration is strongly decreased. And finally, when we treat with an hERG channel blocker, in this case E4031, the field potential duration is strongly increased. So this means if you have a compound where you observe a decrease in the spike amplitude, then most likely this compound is blocking sodium channels. If you observe a decrease in the field potential duration, then your compound most likely blocks calcium channels. And finally, when you observe an increase in the field potential duration, then your compound most likely blocks the hERG channel. So this nicely shows that you can use the change in the field potential waveform to predict which ion channels are affected by your compound. So now I want to switch gears and move on to the Flipper assay. But in order to understand the background of the Flipper assay, it is important to also understand the role of calcium ions for cardiac contraction. So when the membrane is depolarized because of an action potential that arrives at the cell, voltage-gated calcium channels open, and this allows an influx of calcium ions from the extracellular space into the cytosol. This then stimulates ryanodine receptor channels on the sarcoplasmic reticulum, and this allows a further influx of calcium from the sarcoplasmic reticulum into the cytosol. These calcium ions then bind to troponin on the contractile apparatus. This allows actin-myosin cross-bridge formation, and then the actin moves closer to the sarcomeres, which then is the mechanical force for contraction. For relaxation, the calcium is pumped out from the cytosol back into the sarcoplasmic reticulum and out of the cell. And this then allows relaxation and a new cycle can occur. So basically, the intracellular calcium levels in a cardiomyocyte are constantly oscillating. And you can remember that when there are high levels of calcium in the cytoplasm, then the cardiomyocyte contracts. If there are low levels of calcium in the cytoplasm, then the cardiomyocyte relaxes. And so calcium is basically acting as a second messenger, and it links the electrochemical signals of the action potential to the cardiomyocyte contraction. And importantly, changes in the activity of action potential inducing ion channels will also result in a changed shape and duration of the intracellular calcium current. And so for this reason, the intracellular calcium current can be used as a surrogate marker to examine compound effects on cardiac function. So how are we measuring the intracellular calcium oscillations in the cardiomyocytes? We are using a calcium-sensitive dye that can bind to both intra and extracellular calcium. In addition, we are also using a masking dye that blocks signal from any extracellular calcium or extracellular dye. Importantly, the calcium-sensitive dye emits a fluorescent signal that can be captured and recorded. And we are using the Flipper machine to capture and record this signal. Flipper stands for Fluorescent Image Plate Reader. And what is the beauty of this machine is that it can capture and record the fluorescent signal of every well of a plate at the same time and in a kinetic fashion. The protocol that we are using for the Flipper assay is very similar to the MEA assay. Again, we obtain the cardiomyocytes from Fujifilm Cellular Dynamics, and we plate the cells directly into the assay plates that we are using then to capture the fluorescent calcium signal. 7 days after culturing the cells, we add the calcium dye to the cells, and this dye is provided in a kit from Molecular Devices. We incubate the cells for 2 hours at 37 degrees with this dye in order to allow efficient uptake of the dye into the cells. After that, we record a baseline calcium oscillation trace, and then we add the compounds. 15 to 30 minutes after compound addition, we record the post-treatment trace. The parameters that we are measuring for the calcium transient assay is basically used to describe the waveform of the calcium oscillation. And importantly, unlike in the MEA assay, changes in this waveform cannot predict the mechanism via which the calcium oscillation is affected. First, we measure the mean peak rate, or beats per minute, which is basically how many times the calcium spikes per minute. Second, we measure the peak spacing, which is the time that passes between two calcium peaks. And third, we measure the CTD90, or which stands for calcium transient duration, at 90% of decay following the peak amplitude. So this means we are measuring the time it takes for the high calcium levels to go back to almost baseline when only 10% of the lowest levels are remaining. As a proof of concept, we wanted to see how the intracellular calcium oscillation is affected by compounds that block different ion channels, but also that are known to affect cardiac functions via different mechanisms than ion channels. In black, you can see the baseline calcium oscillation in the cardiomyocytes without any compound addition. And then in the different colors, you can see how the oscillation is looking after compound addition. As expected, when we are treating the cells with a calcium channel blocker, in this case we used nimodipine, we see a very big change in the calcium oscillation waveform. In general, we can observe a very strong dampening of the calcium oscillation because we are preventing calcium ions from entering the cell. And interestingly, when we are treating with a sodium channel blocker, in this case we used mexiletine, or a potassium channel blocker, here we used E4031, we can also observe very big changes in the calcium oscillation waveform. And in both cases, we observed decreased beats per minute, increased peak spacing, and increased CTD90. Then we wanted to see how the calcium oscillation is affected by compounds that affect cardiomyocyte function via a different mechanism than ion channels. For this reason, we used a beta-adrenergic agonist. We used isoproterenol specifically, and you can see that this also affects the calcium oscillation waveform. We observed increased amount of beats per minute, decreased peak spacing, and decreased CTD90. And so to confirm that all these changes do not just simply occur with whatever compound that we're adding to the cardiomyocytes, we included a negative control. In this case, we used the COX inhibitor aspirin. And you can see when we treat the cells with aspirin, we do not really see any major changes in the calcium oscillation waveform, indicating that the effects that we saw with the other compounds is specific. Next, we wanted to see if we could see changes in the calcium oscillation with compounds that are known to cause cardiac problems in patients. And so for this, we chose 5 different compounds. We chose astemazole and sertindole, which were both drugs that had been released to the market, but they had to be withdrawn in 1999 because patients who were taking these drugs suffered from TDP and arrhythmia. And then we also chose citalopram, sotalol, and procainamide, which are all still on the market, but they are well known to cause cardiac problems, in particular at higher concentrations. And what you can appreciate from the curves here is that all of these drugs induce a very strong change in the calcium oscillation waveform. So indicating that the Flipper calcium transient assay is very well suited to identify whether a drug might have problems in patients later on. And because the Flipper calcium assay can be performed in the 380-well format, we can treat the cardiomyocytes with a large number of different concentrations of the same drug in the same assay. And so what we have done here is that we treated the cells with astemazole, isoproterenol, and sotalol at increasing concentrations, and we calculate the percentage change for beats per minute, peak spacing, and CTD90 compared to baseline. And what you can see here, for example, that for the beats per minute for astemazole, is that with increasing concentrations of astemazole, you see a stronger decrease in the beats per minute. So meaning that these parameters change in a dose-dependent manner. And that was true for all the parameters that we tested and for all the drugs that we tested as well. So suggesting that we can calculate EC50 and IC50 values of the tested drugs in a very efficient manner. To summarize, what I have shown you today is that an hERG-only focus is not sufficient for drug development. And the reason for this is because QT prolongation and hERG inhibition does not necessarily mean that the drug will cause proarrhythmia in patients. And so for this reason, there is the Cipa Initiative that was initiated in order to improve the accuracy of the proarrhythmic risk assessment by providing a more comprehensive guideline how to assess cardiac risk in new drugs that have been developed. And in particular, the third point of the Cipa initiative focuses on using human-induced pluripotent stem cell-derived cardiomyocytes for in vitro phenotypic assays to assess cardiac liabilities of compounds. And here at Eurofins, we are offering 2 different assays, the MEA assay and the Flipper calcium flux assay, that can be used to evaluate compound effects on cardiac function. So these assays are very well suited to investigate effects of drugs on cardiac function, but they are quite different from each other and complement each other and have different advantages. The MEA assay is an electrophysiology-based technique. So you use microelectrodes that are embedded in the surface of the well to measure fluctuations in the extracellular field potential that is generated by the spontaneously beating cardiomyocytes. And the recorded signal is analogous to the electrocardiogram of patients. And importantly, the changes in the field potential waveform can be used to predict the identity of ion channels that are impacted by the tested drug. And this cannot be done with the Flipper assay. The Flipper calcium flux assay, on the other hand, measures calcium oscillation inside the cardiomyocytes. And the calcium oscillation can be used as a surrogate marker for cardiac function because calcium acts as a second messenger. And the calcium oscillation can also be affected by non-ion channel mechanisms, such as beta-adrenergic signaling, intracellular calcium release and reuptake, etc. And because of the 384 well format, the Flipper assay is also suitable to identify compounds with proarrhythmic risk much earlier in the drug discovery process. So here you can see an overview of the cardiac safety portfolio of Eurofins, which supports drug discovery at all stages. In the earlier stages of drug discovery, you can perform the cardiac ion channel services where you can measure the effect of your drug on 7 different cardiac ion channel currents. And then later on in the drug discovery, you can use the phenotypic cardiomyocytes assay for candidate selection and mechanistic understanding. But as mentioned earlier, because of the high throughput format possibility of the Flipper calcium assay, this assay can also be used much earlier during drug discovery. I thank you very much for your attention. If you want to know more, please visit our homepage for the cardiac safety services, and you feel free to always send me an email. And now I want to hand it over to Victoria for the Q&A session.
Thank you, Verena Albert, for your excellent presentation there. We now have the pleasure to introduce our question and answer session, where Verena will answer all of your questions. Don't forget, you can still submit any questions that you might have using the questions panel, which is in the menu on the right. So now let's take a look at the first question. So Verena, can the results from these assays replace clinical data?
Well, thank you very much for this question. So while the cardiomyocyte assays are much more closely to the in vivo results, they can still not replace clinical data. And that is also emphasized in the Cipa Initiative, where the fourth point is that there should still be done phase 1 clinical trials by focusing on ECG data.
Fantastic, thank you. So what is the turnaround time for the cardiomyocyte assays?
So for both the Flipper and the MEA assay, we have a 20 business day turnaround time, meaning as soon as we have received all the materials, after 20 business days, you should obtain your results.
Great, thank you so much. So next question from our audience now is, do you offer an assay in cardiomyocytes that uses voltage sensitive dyes?
So right now, at the moment, the only assay that we offer using dyes with the cardiomyocytes is the calcium one. If you are interested in voltage, then the MEA assay is the one that you should be performing.
Brilliant, thanks so much, Verena. So next question for you now is, while human-induced pluripotent stem cells express all cardiac ion channels, they often don't express at the correct levels. For example, IK1 current is often lower than expected in mature cardiomyocytes. How do you define the level of IK1 and potentially compensate for immature levels of IK1 in human-induced pluripotent stem cell cardiomyocytes?
Well, thank you very much for this excellent question. And this is maybe one of the major disadvantages of using human-induced pluripotent stem cell-derived cardiomyocytes, as they are not 100% replicating mature cardiomyocytes. So at this point, we do not define the levels of IK1. We obtain the cells always from Fujifilm, so they should have very similar levels throughout our experiments. And we also do not compensate at this point. However, there would be a possibility of, for example, overexpressing this gene in human-induced pluripotent stem cell-derived cardiomyocytes. But what the risk will be then is that these cardiomyocytes lose their ability to spontaneously contract, and then we would need to re-establish the assay that we are currently running.
Great, thank you so much, Verona. And just a quick reminder to still submit any questions that you have using the questions panel. So next question from the audience for you now, Verona. Are we testing antibodies used in oncology in cardiosafety?
So for now, we have mainly focused on small molecules. However, the assay is very well suited to also be testing antibodies.
Great, thank you. So now, what do you think of testing drugs on induced pluripotent stem cell-derived cardiomyocytes from different donors, for example, from different ethnic backgrounds?
This is definitely a very interesting field that we should venture into in the future, because it is very well known that patients with different ethnic backgrounds or different genders react differently to different drugs. So this hasn't been done yet, but I feel like that this is definitely a very interesting field that we should look into more closely.
Definitely fantastic. Thank you so much. Just another quick reminder that you can still submit any questions that you have by using the questions panel in the menu on the right. But next question for you now, Verona. Do you pace the cells in your assays?
So no, we do not pace our cells. So because the cells are spontaneously contracting, and if you culture them the right way, then they are most of the time synchronously beating. So there is no need to pace them.
Brilliant, thank you. So will assays using rodent or high species stem cell derived cardiomyocytes be available in the future?
So as of now, we have no plans of developing any assays where we would use cells derived from non-human species. So the focus that we really have is to provide services where the drugs can be tested for human use later on.
Brilliant, thank you. And what do you think about automated patch clamp on induced pluripotent stem cell cardiomyocytes? Could that add any value?
So this is also definitely a very interesting field that we could venture into, and it will for sure add additional information, as this would allow to measure the action potential in a different way as we are doing now with the extracellular field potential.
Brilliant, thank you so much. So next question from our audience for you now. Since calcium transience assessment is just a surrogate for contractility, is there a risk that we can miss any effects by not using a direct contractility test?
So as with any assay where you use something that is a surrogate marker, I would say there is always a small remaining risk. However, with all the valuations that we have done, we could really see a very nice correlation and observe always changes in the calcium oscillation when we were expecting to see a change. So I would say the risk is very small for that.
Fantastic, thank you. So what is the latest situation regarding regulatory requirements about this phenotypic assay? Will the FDA or ICH guideline be updated to include these assays in the near future?
Yes, so I mean, as of now, the guidelines are still as they are, as I explained, but there has been a lot of discussion that the guidelines should be updated very soon, and that there will be the Cipa Initiative basically implemented by the FDA and other regulatory agencies. So I would expect that this update is coming very, very soon.
Brilliant, thank you. So yeah, thank you for all the fantastic questions that have been sent in. If you do have any, there is still time to send your questions in. But next question for you now, Verena. Could you just remind us of the current recommended testing strategies under the Cipa Initiative?
So yeah, the Cipa Initiative recommends to first test all the new drugs that want to be released to the market on the 7 cardiac ion channels that are relevant for cardiac contraction. Then use this data and perform in silico modeling to predict any cardiac arrhythmia risk. Then as a third step, you should perform in vitro phenotypic assays using cardiomyocytes to see if there are any unanticipated effects, and then finally perform clinical phase 1 studies to see whether you have effects on the electrocardiogram in patients.
Great, thank you so much, Verena. So next question for you, how much do your services cost per compound?
So the cost varies very much depending on exactly how many compounds you are submitting, what kind of frequency. If it is a long-term study, we certainly offer bulk discount, and it also depends on if you're doing the high throughput flipper format or the MEA assay. But you can always reach out to us and we can generate a specific quote for your particular needs.
Fantastic, thank you. And are the assays performed at physiological temperatures?
So yes, all the assays are performed at 37 degrees Celsius, and we try also during all the handling of the cells to keep them at 37 degrees as much as possible.
Brilliant. Thank you so much, Verena, for giving some absolutely fantastic answers. And thank you everyone for sending in some brilliant questions there. Unfortunately, that is all we have time for today, but please do join me in thanking Verena Albert for her time today. As you leave the webinar, a survey will appear on your screen asking you to rate the webinar. Please do take a moment to provide your feedback. If now is not a good time, the survey will be sent to you shortly via email. If you could complete it when you can, we would greatly appreciate it. For now, on behalf of Drug Target Review and Eurofins Discover X, I'd like to thank you all for attending today's webinar. We hope to see you again soon. Goodbye.